See if we can't tie some of this stuff together, which can get very confusing and at first glance doesn't look to be as connected as it actually is. So we've talked about meiosis, talked about Mendel. Meiosis is about making gametes. Mendel is about predicting offspring. And they are very much related. In fact, Mendel's two laws are going to be explained by the process of meiosis. But before we get into all that, let's take a look at what we are trying to examine here. So, the real interest in genetics is to predict traits, to figure out how traits are inherited. Traits are some feature of an organism. Could be hair color, height, skin color, almost anything you can think of is going to be a trait. Now, these human traits are very complex. They're going to follow the rules we're going to talk about, but they're not going to be controlled by one gene with only two different kinds of that gene. So if you want to predict hair color in humans, height in humans, skin color in humans, you can't look at the traits the way we're looking at them in pea plants. And that's part of what was so important about Mendel picking pea plants as his experimental organism. Pea plants reproduce sexually, just like humans. Pea plants had 7 different traits, but those 7 different traits that he looked at, there was one gene controlling the trait, and only 2 different kinds of the gene in the population. So when he looked at pea plants, he came up with the ability to predict these traits in pea plants, which turned out to be the same way that these complex traits are inherited in other organisms. But in other organisms, the difference is it is not one gene controlling the trait, and there are not just 2 different kinds of that gene in a population. But the rules still apply. And by Mendel looking at the pea plant, which turned out to be for these traits very simple, that he was able to figure out what was going on. So before we talk too much more about that, let's get a new page and talk about traits. So traits are descriptions. There are 3 different things we need to keep in mind of when we talk about traits in organisms. And we're going to stick with the simple pea plant traits. So let's actually do that right here. Pea plant traits. So these are descriptions. So the first thing that probably comes to mind when you hear about traits is the phenotype. And the phenotype is literally what it looks like. This is always going to be an adjective. It's going to be something like tall versus short, yellow versus green, whether the buds, the flowers on the plant are at the end of the plant or on the sides of the plant. There were 7 different traits in pea plants that Mendel looked at. And each one of those had a phenotype, and each one of those had 2 possible phenotypes. They were either tall plants or short plants. There were no in between. They were either yellow peas or green peas. They were either flowers on the end of the vine or flowers along the side of the vine. There was no in between. There were 2 possible phenotypes for each of these pea plant traits. What Mendel didn't know, but what he did discover, is that there was one gene controlling these traits. But I have to stress that Mendel didn't know what a gene was. Mendel never used the term gene. He didn't have any idea it was in DNA. It was much later, much later, when it was discovered that these were sequences of DNA written in the form of genes in chromosomes. All he knew is there was one thing controlling whether or not a pea plant was tall or short, yellow or green. And it turns out that we now know that is a gene. The 2 possible phenotypes are because there are alleles of the genes. There are forms of that gene in the population. So there was, for tall, there was a tall allele. And if a pea plant had a capital T gene copy, it would be a tall plant. There was a short allele. And if a pea plant had 2 lowercase t copy, 2 lowercase t alleles, both its copies of the gene for height were lowercase t, it would be a short pea plant. And those were the only 2 options. That's why there were only 2 possible phenotypes. So, with all this, we have not only phenotypes to wonder about, we have genotypes. And genotypes can be both the same, which means you inherited a capital T and a capital T, or a lowercase t and a lowercase t from mom and dad pea plant. And in that case, we called it homozygous. Or you could inherit 2 different alleles. You could have got a capital T from mom, a lowercase t from dad pea plant, or a lowercase t from mom pea plant and a capital T from dad pea plant. And in that case, they were heterozygous. What Mendel found out is that in the case of pea plants, there was a dominant allele for these traits and a recessive allele for these traits. If you have at least one dominant, you get the dominant phenotype. If you have two recessive alleles, you get the recessive phenotype. So if we look over here again at homozygous and heterozygous, the term that Mendel used for homozygous was true breeding. It was true breeding because it had two of the same alleles, two capital T's or two lowercase t's. If it was a true breeding tall, it had two capital T's at the gene for height, and its phenotype was tall. If it had two lowercase t alleles at the gene for height, its phenotype was short, and all of its offspring would be short. No way to get to. What he used for heterozygous can be thought of as hybrids, because if you cross a true breeding tall with a true breeding short, all of the offspring are going to be hybrids. They're going to be heterozygous. They're going to have one capital T, one lowercase t. And then you can cross those to get the ratio of 3 to 1, which brought him to these conclusions that we've been talking about. So the conclusions are that the phenotype of a pea plant is going to be determined by one gene, each phenotype, each trait, one gene, and that gene has two possible forms in the population. Those are called alleles, and one of those forms was dominant and one of those forms was recessive. So, again, we still haven't gotten to the gist of it yet. So for traits, we have phenotypes. These traits are controlled by one gene, and that gene has two possible alleles in the population. True breeders are homozygous, and hybrids are what we get when we cross two different phenotypes that are homozygous, two true breeders. So let's take a look at what that means on a piece of DNA. So this is a chromosome, and we're going to write it in the form of a chromosome, even though we spend most of its time in the form of chromatin. And on this chromosome, about this position somewhere, there is the gene for height. And let's just make it simple. I don't know which chromosome it's on, but we're going to call this chromosome 2. This was inherited from this pea plant's mother, and this one was inherited from this mature pea plant's father. If this is true breeding, there is a capital T allele on this homologous chromosome. So this is homologous chromosome 2. One copy came from the mother plant, one copy came from the father plant. This is an example of a true breeding tall pea plant. Now, over here, we've got a different pea plant. And if we look at chromosome 2 from mom, chromosome 2 from dad, we see that this homologous chromosome 2 is a true breeding short. And this is what Mendel started with. He looked at height, was one of the things he looked at. So he had pea plants that inherited capital T and capital T from both their parents, and over here he had pea plants that inherited lowercase t from both of their parents. So he started with true breeding, which means homozygous for the alleles. He then crossed those pea plants. Now, in the case of an organism, we have two copies of each chromosome, maternal chromosome, paternal chromosome. When you cross something, you make gametes. And those gametes would have two possible forms. The first one, gamete number 1, could have the capital T that came from this pea plant's mother, the maternal capital T. The other gametes would have the capital T that came from the paternal chromosome. So they would both be the same. They would both have capital T's in them. But one of those capital T's came from the maternal chromosome and the other one came from the paternal chromosome. And we could do the same over here with the short alleles. This could also make two different gametes, one with a lowercase t from M and one with a lowercase t from D. They would still be different alleles, even though they are identical as far as the gene goes. So he crossed those, and when he did, let's see if I can fit it here. When he crossed those, all of his pea plants because they got one gamete from here and one gamete from here, had a capital T and a lowercase t, and they all had the phenotype tall. That told him that when he crossed those, he got the 3-to-1 ratio, and that's how he figured out what he got over here. He called these hybrids because they had one capital T, one lowercase t, which leads us to a quick synthesis of all this, I hope. So we saw in meiosis that if you had a chromosome, one of the chromosomes in an individual that had a trait here, we don't have to call it height right now, but one came, this gamete stem cell had two copies of this chromosome. One it inherited from its mother, one it inherited from its father. During interphase, this would be doubled. So now we would actually have four copies of this homologous chromosome. In meiosis 1, that would undergo crossing over and all kinds of other things which don't form tetrads. We don't need to worry about that if we're only looking at one phenotype, one gene. We will in a minute when we look at 2. So at the end of meiosis 1, you had 2 cells. Those 2 cells each had 2 copies of that homologous chromosome. It doesn't matter which ones they're in, but in this case we have 2 copies of the M chromosome in this cell two copies of the D chromosome in this cell, but they're not done. They're both going to go through meiosis 2, and when they do that, we end up with 4 gametes. Let's call these sperm. It's easy to draw those. This one has an M. This one has an M, D, and a D. And at the end of meiosis 2, we have 4 different gametes. Now, if we're only looking at one gene, it doesn't look like we have 4 different gametes because there's M, M, those look the same, D, D, those look the same. But the fact of the matter is, because of crossing over, just because this one has an M and this one has an M doesn't mean they're exactly the same, because parts of the rest of that chromosome came from the D chromosome. And down here, the same thing. Parts of this this chromosome that has the D allele have M pieces on it because of crossing over. So this is where we get Mendel's first law. The law of segregation. So this gamete stem cell has one copy of an M allele, one copy of this D allele. They might be different, they might be the same, but one came from the mother, one came from the father. And the law of segregation says that those two things cannot be in the same gamete. You can't have a gamete that has an M allele and a D allele. The M allele and the D allele have to separate from one another. They have to go to different gametes. And that's true for all the genes. Again, right now we're just looking at one gene, one phenotype. The second law is the law of independent assortment. And that's where crossing over really comes into play. Crossing over says that just because you have the M allele for this gene here doesn't mean all the rest of these are going to be M alleles. All the rest of these are not going to be M alleles. These are not all going to be D. These are not all going to be D. So if you're looking at 2 traits, the law of independent assortment comes into play. Today we're just going to focus on one trait, the law of segregation. So the law of segregation says if you're looking at one trait, the allele you inherited from your mother has to go to a different gamete than the allele you inherited from your father. And that allows us to do what are known as Punnett squares. Let's see. So, Punnett squares. Punnett squares allow us to take advantage of these 2 laws, the law of independent assortment and the law of segregation. Most importantly, the law of segregation. So let's take a look at Mendel's first cross. Mendel took a true breeding tall and crossed it with a true breeding short. So the true breeding tall had a capital T. We're going to go ahead and subscript these. That came from its mom, and it had a capital T that it inherited from its dad. This plant, a different plant, had a lowercase t that it inherited from its mom and a lowercase t that it inherited from its dad. So the gametes that this plant could make would be one of the gametes would have or half the gametes would have the capital T from mom. The other half would have the capital T from dad. Now the gametes that this one could make would half of them would have the lowercase t from M, and the other half would have the lowercase t from Dad. So if we make a little box over here, we can show all the possible ways that the gametes this plant could make could come together with the gametes that this plant could make. So we write the gametes across the top and the side. It doesn't matter whether you put maternal or paternal, either one. And then you fill in these boxes. So in this box, this would have the capital T that this plant got from its mom, the lowercase t that this plant got from its mom. This box, one of the gametes with a capital T from the dad would come together with a gamete with a lowercase t from mom. Down here, we'd have capital T. And over here, and you would see that all offspring are tall, which means they have at least one capital T. That's what it means to be dominant. If you have at least one, you have that phenotype. And they are all heterozygous, which means they have a capital T and a lowercase t. And you can see that they are all actually different because they have different combinations of the M's and the D's. Even though they are exactly the same, they are all equally tall, they all have heterozygous genotypes, but they will have the same phenotype. They will all be tall. So to do a Punnett square, you need to know the genotype of the parents, and you need to know how to make gametes. So for each gene, there are 2 alleles in the adult, 2 alleles in the stem cell in the adult, 1 allele in the gamete. You write them down the sides and across, and that allows you to put them back together. Now, quickly, and if you don't get this right away, it does take practice, and I'm sorry we don't have face-to-face time to practice this, but we can do the same thing if we look at more than one trait. It gets messy if you look at too many traits, but looking at 2 traits is pretty easy. So if we crossed a true breeding tall that had yellow peas, and it was true breeding for that, with a true breeding short that had green peas, we were looking at 2 phenotypes, which means we're looking at 2 genes, and in an individual there are 2 alleles for each, so that means we're looking at 4 alleles total. And all you would do is take this T, combine it with that to give you 1 gamete. The second gamete would be this T could combine with this Y. The third possible gamete is this T could combine with this M, or this T could combine with this Y. I said M before, didn't I? So you have in gametes, instead of 4 alleles, you have 2 alleles for each gene, and they have to be different alleles. You got Ty, 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 Ty. And in that case, your Punnett square would look like this. You would have 4 and 4. These would go down this side. You do the same thing over here, write them over here, and then you fill in each of these squares. Now I'm not going to ask you to do that. I just want to give you a feel for what it looks like. You can do it. The key is to remember that if you have 2 phenotypes, you're looking at 2 genes, and that means in the adult organism, in the stem cell that's going to make the gametes, there are 2 alleles for each gene, and that means you're looking at a total of 4 alleles, and when you make the gametes, you need to put 1 copy of each gene, but only one of its alleles in those gametes, as we see here. And I'm going to cut it there. Like I said, if you can do monohybrid crosses, the one gene, one trait, what Mendel did, you're in great shape. If you do a little practice on this, you can get online and check out other places. Some of the videos will help you.